Have you ever wondered how the blueprint of life, encoded in your DNA, transforms into the physical traits we see in all living organisms? The answer lies within the central dogma of molecular biology, a fundamental principle that illuminates the flow of genetic information within a biological system. The central dogma, coined by Francis Crick, one of the discoverers of the DNA structure, breaks down into three key processes: DNA replication, transcription, and translation. Each of these steps represents a critical phase in the journey from genetic code to functional protein. DNA replication is the process of duplicating the DNA molecule. It unfolds in the nucleus of a cell where enzymes called DNA polymerases unzip the double helix structure of the DNA. Each strand then serves as a template for the creation of a new complementary strand. The outcome? Two identical DNA molecules, each comprising one old and one new strand. Next is transcription, where the code in DNA transforms into a form that can leave the nucleus, the messenger RNA or mRNA. The enzyme RNA polymerase unzips a small portion of the DNA, exposing the genes. It then assembles the mRNA using one strand of DNA as a template. This mRNA carries the genetic message from the DNA out into the cell. Then comes the final step, translation. This process takes place in the ribosomes, the protein factories of the cell. The mRNA serves as a template, and transfer RNA, or tRNA, brings in the amino acids, the building blocks of proteins. Each set of 3 bases in the mRNA, known as a codon, corresponds to a specific amino acid. The ribosome links these amino acids together to form a protein, following the sequence provided by the mRNA. In essence, the central dogma of molecular biology describes the flow of genetic information in cells from DNA to RNA to protein. It's like a biological assembly line where the genetic code in DNA provides the blueprint, mRNA carries the instructions, and proteins are the final product, critical to virtually every function in our bodies. However, it's important to note that while this flow of information is typically unidirectional, there are exceptions, such as in retroviruses like HIV, where information flows from RNA back to DNA. This is known as reverse transcription. To wrap up, the central dogma of molecular biology is a vital concept that elucidates how genetic information passes from DNA to RNA to proteins. It's a process that underpins all of life, from the simplest bacteria to the most complex multicellular organisms, including us. It's a testament to the intricate and beautiful complexity of life, driven by molecules that, though tiny, hold within them the blueprint for all of life's diversity.